Today, we're going to talk about the basics of genetics. These are the fundamentals that you need to know in order to understand how genes are inherited, or in order to know how to use a Punnett square, or how to understand Mendel's genetic experiments using pea plants, for example. And when we're talking about the fundamentals of genetics that we need to know, there are four major things. The first is what a gene is. The second is how genes are passed down from mom and dad. The third is some terminology that we need to understand in order to work with genetic questions. And the fourth is some basic rules about genetics and inheritance. So the first question is, what is a gene? And a gene is a sequence of DNA that determines a particular trait in an individual. Well, what does that mean? Well, what that means is that the DNA that's found inside of all of our cells is really encoding different traits. They can encode eye color, hair color, how tall we're going to be, how big we're going to be, but also the things we can't see, like they encode different proteins and all of the enzymes that are found inside of our body, okay? So when we're talking about genes, we're really just talking about a sequence of DNA that's found inside of our cells. And the DNA is organized into what we call chromosomes. Now, this is known as a karyotype. A karyotype is a photograph of all of the chromosomes that are found in one of our cells. And during certain times, you can photograph the chromosomes in our cells, and then the karyotype lines them up from biggest to smallest. Okay, so what I want you to notice here is these little squiggly lines that we see, those are the chromosomes. And the chromosomes, remember, that's how the DNA gets wrapped up into these structures, okay? DNA gets wrapped up into these chromosomes. The other thing I want you to notice is that there are two of each chromosome. There's two little squiggly lines next to the number 1 because there are two chromosome 1s. And there are two chromosome 2s, and there are two chromosome 3s, and we go along and along until we reach the end. So altogether, there are 23 different pairs of chromosomes in humans. And that means altogether, well, if you have 23 pairs, that means you have a total of 46 chromosomes. Now, one of each chromosome is inherited from mom, and the other of each chromosome is inherited from dad. And that's why we have a pair of each chromosome. So in humans, there are 23 pairs. Now, the 23rd pair, you might have noticed, is made up of possibly two different sized chromosomes. And that's because the 23rd pair, those are special chromosomes known as sex chromosomes. But you can check out a different video to learn about the sex chromosomes. So the second question we have to understand genetics is, how are genes passed down from mom and dad? Because that's where those genes come from mom and from dad. Here's a cartoon, a drawing of that karyotype, right, that shows all 23 pairs. And each one is in a pair, right? So chromosome 9, for instance, there's 2 there. Chromosome 16, there's 2. Each chromosome is actually a pair. You have a pair of them. And one set comes from mom, and the other set comes from dad. And that's why there are 2 of each chromosome. So you can kind of think of it as like, your shoes. You have a left shoe and a right shoe. And if you have 23 pairs of shoes, well, maybe all the left shoes came from mom and all the right shoes came from dad. You have 23 pairs of shoes here, or 23 pairs of chromosomes, but you actually have 46 total shoes, or 46 total chromosomes. So half of the chromosomes come from mom and half of the chromosomes come from dad. And more specifically, one of each chromosome comes from mom or from dad. So where do these chromosomes come from? How are they delivered to you? Well, mom and dad both have cells inside their body that are called germ cells. Now, I know you're thinking like germs, yeah, like bacteria or fungi or things like that, right? No, this germ cell, those are germs. But when we talk about the germ cells found in mom or dad, we're talking about these specific cells that have the ability to undergo meiosis, okay? And these germ cells go through the process of meiosis to become sperm in dad or eggs in mom. Okay, so the germ cells are the only cells that can go through meiosis to form sperm or eggs. Now, we're talking about humans here, right, with sperm and eggs, but really any organism that undergoes sexual reproduction has some form of this process of passing along chromosomes and DNA to the next generation, whether it's plants or animals, humans. Any organism that undergoes sexual reproduction has some form of this process. So these germ cells have two of each chromosome because, mom and dad are human. So all of their cells have a pair of chromosomes, and the germ cells have a pair of chromosomes too. So in this case, I'm only showing you one pair. Maybe this is chromosome 1, and dad and mom both have a pair of chromosome 1. But keep in mind, in those germ cells, there'd be 23 pairs because that's what mom and dad have, is 23 pairs of chromosomes. So meiosis, though, is going to split those chromosomes. It's going to separate the pair so that in the sperm, there's only going to be one of each chromosome. And the same thing for the egg. There's only going to be one of each chromosome in the egg. So just 23, not two sets of 23, but just one set of 23. And that's important if you think about it, because that sperm is going to fertilize the egg. And that fertilized egg is the first cell that becomes a new individual. So the sperm is going to contribute one set of 23 chromosomes to this human, and the egg is going to contribute the other set. And together, that's going to have two of each chromosome, or 46 total chromosomes. And that's what it is to be human, is to have two of each chromosome and to have 23 pairs, or 46 total chromosomes. We call the sperm or the eggs gametes. And just as a side note, sometimes you'll hear spermatozoa, and that's the same term as sperm. And for eggs, mom's eggs, they're called ova, right? And a singular egg, one egg, is known as an ovum. So you might also hear those terms for mom's eggs. But the sperm and the eggs, those are the gametes. So germ cells undergo meiosis to form gametes. And in dad, that's going to be sperm. In mom, that's going to be eggs. Now, these germ cells are found in the testes of men. That's where meiosis of those germ cells takes place to form the sperm, which are also found in the testes. In mom, those germ cells undergo meiosis in the ovaries to form the eggs. So in the process of fertilization, we have a sperm that's going to enter an egg. And the sperm is bringing one set of 23 chromosomes. The egg has the other set of 23 chromosomes. So together, that fertilized egg has two sets, right, of 23 chromosomes. It has a pair of each chromosome for a total of 46 chromosomes. Now, that fertilized egg is also known as a zygote. So scientifically, when sperm enters egg, when fertilization occurs, now we have a zygote. So here's a fill-in question for you, just to see if you can bring all this terminology together. Pause the video and see if you can fill in words in the 4 blanks that are found here. Okay, pause the video. Okay, did you pause the video? All right, well, let's talk about this and see if we can fill in the different terminology here. Okay, blank undergo meiosis to produce blank in men and blank in women. Okay, germ cells are the things that undergo meiosis, and those germ cells are going to differentiate or become sperm in men and ova or eggs in women. Just as a reminder, here's that diagram we used where the germ cells in dad are going to undergo meiosis to form the sperm, and the germ cells in mom's ovaries are going to undergo meiosis to form her eggs, her ova. Now, here's the second sentence. When fertilization occurs, so when a sperm enters an egg, a blank is formed. And in this case, it's a zygote, right? A fertilized egg, which is scientifically known as a zygote. And here's the process, right? We have 2 different kinds of gametes. There's sperm and egg. So when sperm enters egg in the process of fertilization, a zygote is formed. And that zygote now has the full set of chromosomes. 23 from mom, and 23 from dad. So here's another graphic just to sort of remind you that there are 23 individual chromosomes coming from dad and is going to be found in dad's sperm. And there are 23 individual chromosomes that are coming from mom in her egg. And so when the sperm fertilizes the egg, we end up with that zygote, that fertilized egg that now has 2 of each chromosome. So the process of meiosis is really important because if those chromosomes didn't get separated for each pair, then when fertilization occurs, there would be 4 of each chromosome inside that fertilized egg. And that's not conducive to life. That's not going to cause a cell that, becomes an individual. There's some terminology we use to talk about the gametes, the sperm or the egg, or the other cells in our body. And the gametes are known as haploid. Okay, haploid is a term that we use to indicate that that cell has 1 of each chromosome, not 2. Okay, so the sperm and the egg only have 1 of each chromosome. They're haploid. All other cells in the body are known as diploid. And you can remember that because di means 2. So diploid cells have 2 of each chromosome. And here's an easy rule to remember. In humans, all cells of the body are diploid, except for the sperm or the egg, except for the gametes. Okay, the gametes are haploid. So all cells in your body are diploid. They have 2 of each chromosome, except for the sperm and egg, which are haploid. They only have 1 of each chromosome. Let's talk about some terminology that we're going to need to understand in order to work with these genetic problems. We learned about what a gene is. Gene is just a sequence of DNA that's going to encode a certain trait. And now when we think about it, those genes are arranged within a chromosome, right? Because the DNA within our cells gets packaged and wrapped up into this tighter structure called a chromosome. That's really important. If you think about it, we have about 2 meters of DNA. If you were to lay it end to end, we have 2 meters of DNA in each of our cells. They have to be wrapped up into something more compact so that they'll actually fit within the cell and not just be like a jumbled mess. Okay, so you can kind of think it like string, right? If you pulled all the string off of a spool, it would just be a jumbled mess. But the spool allows you to wrap it up so that instead of taking up this much room, it takes up this much room. And that's what the chromosomes are, right? The DNA gets wrapped up into chromosomes that are tighter and more compact and orderly. So those chromosomes are where our genes are found because that's where the DNA is found. So what I have here is just a drawing I made of 1 chromosome. And I put 3 different genes on it, gene A, gene B, and gene C. Now in humans, we have about 20,000 different genes. So in actuality, every chromosome is going to have more than 3 genes. But in this case, we're just looking at 3 genes, gene A, gene B, and gene C. Now, the human cells, right, that zygote, that fertilized egg, is diploid. So it has 2 of each chromosome. So that's why I've doubled it here. 1 of these chromosomes came from mom, and 1 of these chromosomes came from dad. So the way that we describe the pair of chromosomes is we call them homologous chromosomes. Homo means same, right? So these are the same chromosome. But that doesn't mean that the genes on them are going to have the same versions. We see here that the chromosome from mom has genes A, B, and C on it, and chromosome from dad is the same chromosome. It's the homologous chromosome, so it has the same set of genes as well, genes A, B, and C. But the versions that got passed down on these chromosomes might be different versions. Let's look at gene A, for instance, right? Gene A, well, maybe this gene encodes eye color, and dad passed along the version for blue eyes, and mom passed along the version for brown eyes. So it's the same gene. The gene A, is the gene that's going to encode eye color. But the version that gets passed along is not necessarily always the same because we have different eye colors out there, right? So the different versions might not be the same on both chromosomes. And so we use this word called allele to indicate the version that's present on the gene. Allele. So go ahead and say that word because that's sort of a tough one if you're just looking at it to how to pronounce it. Allele. Allele, right? Like Muhammad allele. Okay, that can help you remember how you start off that word. Allele. The alleles are talking about the different versions. So dad passed along a blue eye allele, mom passed along a brown eye allele. Those are the different versions of a gene. If we look at gene C, well, mom and dad passed along the same version, the same allele. Okay, so in this case, an individual would be homozygous. Okay, so homozygous means that the person received the same allele from mom and dad. Now let's just take a look at these two words that have homo. Homo means same, right? But when we're talking about the chromosomes, we're talking about homologous. Okay, like a homolog is sort of the same, right? So homologous chromosomes, we're talking about the two chromosomes together, same chromosome, chromosome 1, chromosome 1. Homozygous, we're talking about the gene version that came. And if you look at this word, homo means same, but zygous should remind you of zygote. That's a fertilized egg. So we're really talking about, well, that fertilized egg, which version of the gene ended up in, which versions of the genes ended up in that fertilized egg, in the zygote. And if that fertilized egg ended up with the same gene version from mom and from dad, we call it homozygous. Okay, same gene version. If we look here at gene B, well, gene B is like gene A, and that there were two different versions that were inherited in this individual. Let's say this is for hair color. So dad passed along an allele for red hair, and mom passed along an allele for brown hair. In this case, they have two different alleles. And so in this gene, this person is heterozygous. Hetero means different. So they ended up with two different alleles passed along from mom and from dad. And so let me ask you here, gene A, would that be homozygous or would this person be heterozygous for the alleles on gene A? All right, gene A, there's two different versions, blue and brown. So because they have two different alleles, that person would be heterozygous for the eye color gene. See how that works? So all this terminology, put it on note cards, put it on your phone so you can come back to it and practice it, because that's the only way that we get better at it, is by memorizing what the terms mean and then by using the terms. So when we're thinking about the pair of chromosomes, the homologous chromosomes, one of each chromosome, each of the 23 chromosomes is carried from dad's sperm, and the other set of 23 chromosomes comes from mom's eggs. And so when fertilization occurs, that zygote or fertilized egg now has two of each chromosome. So they have 23 pairs. And again, the pair are called homologous chromosomes. So chromosome 17 from mom and chromosome 17 from dad, those are homologous chromosomes. And the same thing for all the other pairs. We call all of the pairs homologous chromosomes. So the most important thing is that we have to talk about some basic rules about genetics. Now let's consider, when we're talking about the basic rules, we're going to need to use that terminology: allele, heterozygous, and homozygous. Here are two chromosome examples that have 3 different genes on them. And so if there are 2 different versions or alleles present, then one is dominant over the other. So in the case of the first allele, A, right, we have 2 different alleles, and gene A encodes eye color. We have a blue allele and we have a brown allele. So one of those is going to be dominant over the other. Well, what does that mean? The dominant allele is the one that is going to be expressed. And when we're talking about something being expressed, it means that that gene is going to be turned on. We're going to be able to see the effects of that gene. The recessive allele is not going to be seen. Okay, so the dominant one sort of overrides the recessive one. The only time you're going to see the recessive allele expressed or seen is if we have 2 of them. So in this case, we have a brown eye allele and a blue eye allele. One of those is dominant over the other. And so we have a dominant allele and we have a recessive allele. Now, when we're talking about the dominant and recessive alleles, the convention is that we're going to write the dominant allele in an uppercase letter. The recessive allele, we're going to use the same letter, but it's going to be in lowercase. Now let's work through some of this, okay? So here's an example, and let's use gene 1 for eye color. Okay, so when considering the gene for eye color, the brown allele is dominant over the blue allele. So we're going to use capital B to indicate the brown allele, and we're going to use lowercase b to indicate the blue allele. Now, why did we pick B? We picked B because the dominant allele is brown, and brown starts with the letter B. So we're going to use capital B for the dominant allele, for the brown allele, and we're going to use the same letter, B, but it's going to be in lowercase to represent the recessive allele. So we're going to use big B for the brown allele and little b for the blue allele. Now, I know it's a little confusing with this example because blue and brown both start with B, but the letter we generally choose is going to be the first letter of the dominant allele. And so we're going to use that uppercase letter to indicate the dominant version and the lowercase letter to indicate the recessive allele, okay? Now, if we have a brown eye allele and a blue eye allele out there, they can come together in 3 different ways. We can have someone who has 2 dominant alleles, 2 brown eye alleles. And so because brown is the dominant allele, we're going to indicate that this person is big B, big B. They have a big B allele, a brown allele on one chromosome, and they have a brown allele on the other chromosome. Now, in the population, we also could have someone who inherits a brown allele and a blue allele. So big B, little b. Big B stands for the brown allele, little b stands for the blue allele. And then there are people in the population who could have 2 recessive alleles, 2 blue eye alleles. So that would be little b, little b. So we have the words homozygous and heterozygous, but we can specify that a little bit more when we're talking about the different possible options out there. So big B, and big B, little b are homozygous because they have the same version. But big B has 2 of the dominant allele. So we call that homozygous dominant. Whereas little b has 2 of the recessive allele. So we call that homozygous recessive. So right away, if I tell you that somebody is homozygous recessive, you know they have 2 of the recessive alleles. Or if I tell you that somebody is heterozygous, then you know they have 1 dominant and 1 recessive allele. Now, there are 2 other important terms that are related to this, and that's genotype and phenotype, because our genes are going to be expressed, and that's what we can see, right? So when we're talking about homozygous dominant, big B, we call that the genotype. All right, so the genotype is the type of gene version that they have. So big B, little b, little b, that's their genotype. The genotype could be homozygous dominant, heterozygous, or homozygous recessive. We're talking about the genes. Those are the genotypes. But the phenotype is what you can see, right? So big B is not the phenotype. We don't see the genes. That's the genotype. If somebody is homozygous dominant, what color eyes will they have? That's the phenotype, okay? So the phenotype is the expression of those genes. It's what we can observe. So let's walk through these and figure out what the phenotype of each possibility is, okay? So for someone whose genotype is big B, what is their phenotype going to be? Phenotype's what you see. We're talking about eye color. And because they have 2 dominant brown alleles, well, their phenotype is going to be brown eyes. How about big B, little b? What's going to be the phenotype of somebody with a big B, little b genotype? Right? Their phenotype is going to be that they also have brown eyes because they have a dominant allele and a recessive allele. But remember, if you have 1 of each, the dominant one wins. And we'll see the dominant one. That's the one that ends up being expressed. Okay, how about somebody with a genotype that's homozygous recessive? Little b. Right, so little b means that they have a blue eye allele and another blue eye allele. So where that individual's phenotype is going to be that they have blue eyes. Okay, because there's no dominant allele, there's two recessive alleles. And that's the only way that someone's phenotype is going to be the recessive phenotype. The only way that you're going to see blue-eyed individuals is if they have two blue eye alleles, because that's the recessive allele. So the cool thing about this is that you might have two people with the same phenotype, but they might have a different genotype. And this is a great example. You could have two people with brown eyes, right? That's their phenotype. But this person might be homozygous dominant, big B, big B, and this person with brown eyes might be heterozygous. They might have a recessive blue allele as well. It's just being masked by the dominant brown allele that they have. Let's do another example. Okay, here's example 2. This example deals with the shape of a person's thumb. And so when you look at the gene for thumb shape, the allele for a straight thumb is dominant over the allele for what we call a hitchhiker's thumb. Now, I happen to have a straight thumb, okay? So that just means that my thumb kind of goes straight up and it points upward when I do like, a thumbs up sign, right? It goes straight up. Now, there's another version where the top part of the thumb bends backwards towards the arm, and that's lovingly known as a hitchhiker's thumb when that version bends backwards, okay? Well, the allele for a straight thumb, that's the dominant one. The allele for the hitchhiker's thumb is the recessive allele. So in this case, the dominant one is the straight thumb. So straight, we're going to use the letter S. So capital S is going to refer to the dominance allele for a straight thumb, whereas lowercase s is going to refer to the hitchhiker's thumb. So when we think about the population, right, everyone's going to have 2 of each chromosome. So they're going to have 2 alleles for this particular thumb-shaped gene. They could have a big S and a big S allele, one from mom, one from dad. They could have a big S and a little s allele, or that individual could have received the recessive allele from both mom and dad. So this person could be little s, little s. Big S, that's homozygous dominant. Big S, that's heterozygous. And little s is homozygous recessive. Now, again, let's take a look at the genotype and the phenotype. So the genotype, someone with a genotype that's homozygous dominant, let me ask you, what will their phenotype be? An individual that's homozygous dominant, that's their genotype, what will their phenotype be? Okay, well, if we look at the information, homozygous dominant means that they have 2 of the dominant alleles. The dominant allele encodes a straight thumb. So someone's phenotype would be that they have straight thumbs. A person that's heterozygous, they also would have the straight thumb phenotype because they have a dominant and a recessive allele, and the dominant allele wins out. So straight thumb because that's the dominant allele. A person whose genotype is little s, their phenotype is going to be that they have a hitchhiker's thumb because they have 2 of the recessive alleles, and without any dominant allele there, it's the recessive allele that's going to be expressed, and the recessive allele encodes the hitchhiker's thumb. See how this works? So just to summarize everything up so you can bring it all together, and maybe even pause the video here and see if you can explain these or at least define what all this stuff means. We talked about what a gene is and how genes are inherited or passed along from generation to generation. We talked about what meiosis is and how meiosis is the process by which germ cells become either sperm in males or ova, eggs, in females. We talked about the process of fertilization, where one sperm enters one egg to form that fertilized egg, that zygote. And now that zygote is diploid because it has 2 of each. And then we also talked about how sperm or egg, they're haploid. They only have 1 set of each chromosome. But all other cells in our body, including zygotes, are going to be diploid, or they're going to have 2 of each chromosome. We also talked about what the word allele means. That's the different gene versions. We talked about how when there's different alleles, one is going to be dominant and one could be recessive. Now, it's possible someone receives two dominant or two recessive, but if they have one of each, then the dominant one is going to win over the recessive allele. We talked about how we typically try to write the dominant or recessive alleles so that we can understand which is dominant and which is recessive. And we talked about how they can be homozygous, right? That someone could have two of the same version, but they could be homozygous dominant or homozygous recessive. They could have two of the dominant alleles or two of the recessive alleles, or if they have one of each, that's called heterozygous. We also talked about what genotype and phenotype is, where genotype refers to the genes that we're talking about, and phenotype refers to the expression of those genes, what we see. So these are the basics that we have to understand if we're going to, for instance, use a Punnett square to show how genes are inherited, or maybe understand Gregor Mendel's experiments with pea plants that ended up becoming the foundation of what we know today as genetics. Or these are the basics that we need to understand if we're going to work with human genetics and how human genes are passed along and inherited.